Welcome to the Investor Download. I'm your host, David Brett. With me in the studio is Harry Rakes, Investment Director. Hi, Harry.
Hi, Tim.
How are you doing?
Good, thank you.
And we've got a very special guest down the line with us today. He's Gordon Sanghera, co-founder and CEO of Oxford Nanopore Technologies. Gordon, a very warm welcome to the show. How are you doing today?
I'm good. Afternoon, everyone.
And where do we find you today?
I'm actually at our Oxford offices, which are just off the Ring Road, in about 3 miles from the city centre.
You make it sound very glamorous.
It is. Next to a water waste disposal site.
It doesn't get much more picture perfect than that.
Sewage works.
Well, thank you so much for joining us.
Which actually is very interesting.
Go on, sorry.
When you think about DNA sequencing, which is what we're going to talk about later.
Yeah, absolutely. So you're here today to talk about, well, specifically your company, but more broadly, the industry in which you work. So I thought we'd better start off with a rather basic question, because there'll be a lot of people that are either watching or listening in today that won't know much about the company itself. Can you just give us a background as to what Oxford Nanopore is? Yeah,
absolutely. So Oxford Nanopore was founded, actually, let's go all the way back, in 2005 as a spin-out from the University of Oxford. And the technology we acquired from the university was a new method of measurement, and that's electronic single molecule measurement. And the company, as I said, has been around for 18 years. We were private until October 21, when we floated on the London Stock Exchange. And today, and for the last 7 years, we've been shipping products, nanopore DNA sequencers, we'll talk more on that later, to over 125 countries. And we launched products in 2015.
And how did you become involved with Oxford Nanopore?
So I had been, prior to joining Oxford Nanopore, I'd been with another Oxford spin-out. It was a company called Medisense, who had developed electronic glucose sensors for home testing. So if you know any type 1 diabetics, they need to keep constant check and control of their glucose, primarily through insulin injections and measuring glucose frequently. And that's coupling biology to electronics. And that company was spun out of Oxford in the mid-80s, showing my age now. And in 96, it was sold to Abbott Diagnostics for $876 million. I spent 7 years at Abbott before starting to get itchy feet, and decided to try and see if there was another big adventure in me, and came across Oxford Nanopore. And that's kind of how the whole thing moved on. Oxford Nanopore is very similar technology, very different application areas, blood glucose testing versus DNA, but very similar underlying platform connectivity. And I was quick to see how we could scale a very academic experiment into potentially an easy-to-use platform for measurement.
Okay, so can you describe, in as simple terms as possible, DNA sequencing, which is what Oxford Nanopore is all about?
Yeah, so why is DNA important? DNA is a source code of all living systems. And each of us, humans, have 3 billion letters in one continuous string of GTAC, so it's a 4-base code. And we are interested in how are we different, how that uniqueness responds to predisposition to disease, efficacy of treatments, and everything. And it's not just humans, us, it's the environment, the food around us, and pretty much DNA is a source code of all living systems. So being able to interrogate and understand how our source code is evolving and changing over time in reaction to our environment as we age is really important, and underpins our understanding of how to monitor and diagnose diseases, whether we're thinking about human health, plant pathogens, environmental disasters we see from time to time. DNA sequencing is the method by which we read these source codes. Today, that methodology is primarily dominated by large centralized facilities with multi-million-dollar instruments. So if you like, DNA sequencing, or the access of our source code today, is somewhat akin to mainframe computing in the early 70s, early 80s, late 70s. And we at Oxford Nanopore have developed a series of platforms that will bring about a much more affordable, accessible, low-cost way of accessing DNA RNA information in the same way as we saw the revolution from mainframe to desktop to handheld computing in your iPhone today.
I was going to say, I should suspect most people like me first came into contact with DNA or the idea of DNA in Jurassic Park. And I'm assuming quite a lot of that, or the methods about getting the DNA, have changed quite a lot over the last almost 40 years. How has it changed?
So in Jurassic Park, there's that iconic scene where they show live streaming of DNA, and it's in this particular case, I think it's one of the dinosaurs. That's pretty much what we do. So moving DNA sequencing from large multi-million-dollar batch-based systems, so you have to maximally load these machines in order to get economies of scale, and it takes multiple days to prep these samples, multiple days to run the system, and then multiple days and infrastructure. So large infrastructure at the front end, $1 million for a sequencing machine, $250,000 to $1 million, and then multi-million-dollar IT infrastructure to do back-end software analysis. What we do is we have this direct single molecule electronic measurement platform, and we can literally take that iconic double-stranded DNA and singulate it and send it down a very small hole. The company's name is Nanopore, very small hole, and read it directly, very simply. That means your sample prep goes from days to minutes. The equipment and our cheapest sequencer comes in at $1,000.
Oh, wow.
Here's one here. Let's just get it on camera, sorry. Okay, we're nearly there.
Oh, wow, that is small.
There we go.
Yeah, look at that.
So that is, it's about the size of a Mars bar. And inside it, let's see if I can get this done. We have what we call a flow cell, and that's our consumable. So the business model is razor blade. Now, the disruptive commercial model we have, I'm going to put that down, is that we can provide these instruments for free. So affordable and accessible. You no longer need a multi-million-dollar CapEx budget. And they stream that DNA information live in real time. And they're not batch-based. You can run one sample for half an hour. Maybe they're a small virus, which is 10,000 bases, typically in number of DNA bases on that string. Or you could run it for multiple hours. We might be looking at bacterial pathogens, which are 3 to 5 million bases, or much longer for human, which is 3 billion, or plants, which run into tens of billions. So you absolutely change the value proposition from maximally loading a very expensive machine in order to get economies of scale to being able to do real-time, fully accessible information streamed live. Now, what's important about that, and the analogy to the computer revolution, is that the minute we were able to provide real-time data at your fingertips, it ushered in the information age, and that was catalyzed by the computer, the personal computer. That affordable accessibility to allow people to get that data and information in real time. Our platform is highly differentiated from those large mainframe sequencing platforms, and we can provide real-time DNA streaming of information in an affordable, accessible manner, which was particularly important during the pandemic.
I was about to say, I was going to ask you for some real life examples where this technology has helped you change things going forward. Can you bring it to life for us?
Yeah, and this is not just a pandemic platform. In fact, it's the complete opposite, but it's a nice way to describe why DNA sequencing is so important. As we all saw, a new species emerged, SARS-CoV, and the way it mutated and changed over time, and was it a variant? Did it have mutations that were variants of concern from a health perspective? Was it more virulent? Was it less virulent? All these things come from the continual changing of the biology of something like the COVID virus. And it's really important to understand how these changes track and trend, and whether they become benign or more dangerous to us all. And so it became clear, I think in some respects, biology, mutational biology 101 was learnt by everybody as a result of the pandemic. And that's a really good example of how public health laboratories adopted our platform. And over 85 countries and 2 million COVID genomes were sequenced. So full genomes read on nanopore sequencing. What's quite significant within that stat is that a lot of them were in low and middle income countries. So for the first time, we are seeing countries that don't have tens of millions of dollars to build infrastructure to have sequencing power being able to enter the game. And there is going to be another pandemic. There's no doubt about that. And having these distributed, decentralized, real-time surveillance networks is how we are going to prevent the next pandemic. But more broadly, it illustrates why it's important for us to interrogate and understand DNA sequencing of the things around us, the things in us, our own bodies, and everything living.
And from what you're saying, I'm going to guess that probably going forward, the next pandemic is going to change, this type of technology is going to change the way we attack the next pandemic. It's not going to be done in the same way we've done it this time around.
So it's interesting, post-pandemic, there's a lot of talk about how we prevent the next pandemic, and there is a lot of networks, Pan-Asian network, for example, the China CDC, Japan, who are putting out distributed centers of excellence using nanopore sequencing so that they can be continually monitoring and looking for anything that's unusual. And when you see that, you're alerted more quickly. Now, in order for that to work properly, that's going to have to be a global network at some point, and rather than pockets of good practice, which is what we're seeing right now. And there is a lot of talk about how we prepare for the next pandemic, and it will need that distributed capability to read all the way from low and middle income countries right through to the rich superpowers very quickly, these changes and how it's spreading. So yes, I think it will be a game changer.
That seems like a massive undertaking. I presume you're speaking to quite a lot of countries and heads of countries about this going forward.
It's a mixed bag. There are some, I mean, the Pan-Asian network is great. It's based out of Singapore, and you've got the Asia-Pac region all pulling together. Funnily enough, less so in Europe and North America. I guess because we do have the centralized capabilities. we're less exposed. And yes, it'll take time, but I think it will happen and is happening, because the value proposition is very cheap, very affordable, and very accessible. And I think that will bring that change about over time. But you're right, getting the whole world to do something as a collective is very challenging.
Yes, we've tried and failed before, but hopefully next time round we'll all come together. Harry, you're nodding away. I mean, from an investor's point of view, this must be quite an attractive story.
Absolutely. I mean, what Oxford Nanopore has been doing is at the cutting edge of innovation in its sector. These are kind of themes that we're looking to get exposure to as investors. As Gordon said, he's been with this business since 2005. He knows the sector inside out. And so it's investing with teams like Gordon and the team at Nanopore to really target this really exciting opportunity.
Gordon, I just want to, if we may, just compare how your company goes about DNA sequencing compared to other companies, because there'll be a lot of people out there that won't understand the difference.
Absolutely. So the simplest way to think about what we do is we are shifting from the way sequencing is done today, and I'll explain a little bit in a minute. You end up taking the beautiful, rich content and color imagery of biology and turning it into a very dull black and white image. Because of the way the current systems work, they make a photocopy of this beautiful color, full biology in full color, into a very grainy black and white picture. And that's just how the sequencing technology works today. And that means you really wash out quite a lot of important biology and miss very key drivers of many diseases and changes that occur. What we do is we actually, as I've said, we take this double strand and we singulate it and we send it down a hole, and that hole is 1/10,000th the size of a human hair, just to give you a sense of scale. But it's the hole is a naturally occurring protein, which are doors into living cells. So we take that biology, that cell biology, out of a living system, and we put it into an electronic measurement platform. As we send the DNA down the hole, we are looking at native, complete, high definition, full color DNA. So we can see all the things that you miss with your grainy black and white picture. And this is the first time since we launched our products in 2015 that we have started to read biology in full high def and full color. The richness of content that comes from that is really opening up areas of the dark genome, which today probably account for over 1/3 of genetic drivers of disease that you cannot see today. And so it's not just that it's smaller, faster, cheaper, affordable, accessible. It's that you're reading the native DNA, and that's hugely important. If I just give you one simple example, it is widely accepted and understood the smoking gun for cancer predisposition is, and even treatments and efficacy of treatment, is something called methylation. And that's where your GTAC, your C base, is chemically corrupted. And that's called methylation. And that methylation pattern is a result of the environment, your lifestyle, and that is a real driver on predisposition to cancer and how certain treatments will work for you. So these modifications aren't just pretty color things on the edges and fringes. These are key drivers of diseases like cancer, and understanding and seeing biology in full high definition color is a really exciting moment for the industry.
Yeah, it sounds like you're almost going from a 2D view of things to pretty much a 4D view of things, right? 4 dimensions, you get the whole package.
Indeed. And there is in the 7 years we've been selling, there's a growing re-understanding, if you like, of the biology, as they can see this much more comprehensive richness of content that's not possible with the current systems.
And this richness of content, what potential impact might that have on, I don't know, medical research and diagnosis? You mentioned cancer there, obviously.
Yeah, so I mean, so actually the interesting one, I think, that is really getting traction quickly is hard to diagnose, essentially newborns, but it can be all the way up to adults, because they've been on a diagnostic odyssey where they've not been able to find the underpinning cause of disease. So these are people with known family history of genetic disorder, and we're really seeing progress in really shedding light on those hard to diagnose newborns. Essentially, that's where it's starting. At the other end of the spectrum, developmental disorders in neurodegeneration, such as Alzheimer's, because our technology is capable of shedding light in these dark corners that you cannot with existing technologies. There is a real correlation between something that's referred to as structural variations. So that's where maybe 100 or several thousand of your DNA letters in that 3 billion string are in the wrong place. So a structural variant. So it's many, many bases. And these are known to be linked to neurodegeneration. So we're starting to see early indications of those correlations now that we can see this more clearly in neurodegeneration, in cancer, which is early, but in direct impact in medical care, coming very quickly with people with developmental disorders and known family histories of hard to diagnose genetic diseases.
It sounds like a complete game changer, both for the people that have those diseases and for the families that have to care for them.
It will be, and this must be caveated because it takes time to revalidate what we already know. So the current single point mutations, this SNPs and SNVs, single nucleotide polymorphisms and single nucleotide variants, there is a very large database and correlations that underpin disease. We're now adding in all these other pieces, and that does require time to revalidate the genetics linked to particular diseases. So it's very exciting, no doubt about that, but we're in this technology validation phase from which we will start to see that sort of clinical routine utility coming over a matter of time in the medium term.
And how accurate and reliable are nanopore technologies in DNA sequencing?
So the basic underlying technology today is equivalent in accuracy to the existing mainframe systems, if you like. As with most disruptive technologies, whether you think about the initial mobile phones and their quality and their range, or digital photography and the pixelation and the quality there, we have been on a journey. We launched the product when it was good enough, and over the last 6 or 7 years, we have continued to improve and enhance the technology such that we are now substantially equivalent, and we can catch all of these single point mutations that the existing technologies have, but we can also add so much more content and color. And so we're now at a point where we are equivalent and actually moving past the existing technologies because we provide so much more richness of content.
And you mentioned you're in the validation process. Are there any limitations at all or challenges associated with the technology?
Limitations. There will be limitations. I think we're in a phase of, it's the complete opposite. There are so many things and so many publications coming out from researchers. It's a very broad opening up of potential applications. In terms of limitations, that's a hard question to answer. It certainly can do more than the existing technologies, but there's always going to be a limitation. But we have managed to get it onto the space station, and it's running up on the space station as we speak, and NASA are going to take it, hopefully, on the next mission to Mars. So it's doing okay so far. It's a very robust, rugged technology. And I think there's an interesting opening up of what this technology could do, and beyond human health as well. It's always easier when I do these kind of talks about the company to talk about human health, and it's easy to sort of get people to understand that, but there's so much more that we can do.
Gordon, I was going to ask a question associated with what David just asked about, the challenges or any sort of limitations of the technology. But as a sort of challenger in this space, educating people that are using this technology, going up against a big incumbent, how have you found that process over the last sort of 7-8 years that you've had a product in market? Educating the market, really.
It's been tough. I mean, there's no doubt about it. There's one market leader, Illumina, and which, by the way, was a UK Cambridge acquisition of a spin out. So the underlying sequencing chemistry was invented in Cambridge. The whole field for the last 15 years has been dominated by one monopolizing market leader. So it makes it very difficult to topple them because they are such a big, powerful voice. And I think I was surprised, have been surprised how hard it has been. And what I've realized in the last 12 months is, in order to move past Illumina, the customers out there today need to be able to see exactly what they already see with their existing system. And that sounds very obvious now, but I was surprised by that. My expectation was that once we showed them so much more and all this richness of content, they would move towards our technology. But it's been absolutely clear that you need to be able to give them what they already get from their existing systems and then add value. And that's something that's kind of happened since we launched our latest kits and upgrades, which was a year ago now. So, but it's still very challenging to get that, voice heard in a single, monopolizing, market-leading, dominant player. And that's why some of the low and middle-income countries adopting the technology has been a rich source of growth for us, and why Asia-Pacific is quite important to us. Of course, in Asia-Pac, we have BGI, which is the Illumina equivalent. So there are two very big beasts that we're taking on. But all the features and benefits we have, people will vote with their feet over time. It's taken longer than I thought it would to get commercial traction and acceptance, because there is one dominant, clear voice out there.
Gordon, you mentioned markets there. Unlike some of your peers, you decided to list in the UK. In fact, a lot of companies in this country, in the UK, have decided to list in New York as opposed to London. Why is it you chose to list in the UK rather than on the NASDAQ in the US?
It's a good question. I'm asked it quite a lot. When we did the analysis with our advisors, London versus NASDAQ, given the center of gravity of this company is here, the management and the executives and board are very UK based. We have a very strong share register, particularly some of our earliest investors, high quality London based. The overall analysis, it was quite marginal London versus NASDAQ. And given that we had been up to that point 16 years private, to move from private to NASDAQ with quarterly reporting and very tightly regulated, highly compliant markets was quite a big leap. So on the basis of all of those pieces, we decided to stick with London. And the main question we asked was, will it prevent people globally from investing? And the answer is no. And particularly, will traditional NASDAQ investors invest in the LSE? And the answer is yes. So we've managed to attract the right sort of shareholders, and something like 40% of investors came in from the US at IPO, and it was on the basis of that. As we're in this explosive growth phase, we strongly felt that quarterly reporting would be a real challenge while we're really trying to grow the business at pace. And twice a year is a good cadence, and we've been able to cope with that and scale the corporate functions to match the requirements of public versus private company.
Okay, let's throw it forward a bit. I mean, how do you see DNA sequencing and the whole genome sequencing evolving over the next decade?
So it's interesting that today we are 20 years on from the first human genome that was mapped in 2003. 20 years on, last year on the front cover of Nature, long read sequencing, nanopore sequencing, was featured as the method of the year. And the final 8% of the genome that had been hidden or dark was mapped last year. So now we have a fully annotated genome. That 8% doesn't sound like very much, but it actually accounts for over 1/3 of diseases. So when we start to think about the implications of better understanding neurodegeneration, earlier diagnosis of cancer, rapid real time, and this is going back to the Jurassic Park example, sequence data being made available in, for example, intensive care, where you have respiratory infections that are mutating in real time and getting results in 4 hours rather than 4 days. All of these will be game changing. But and that's in the next couple of years. In answer to your question, in the next 5 to 10 years, the ability to be able to sample, access, and read DNA in real time will be as big a shift as we saw with the capabilities and the revolution of the information age. So we're entering the genomic information age, and whilst the Internet of Things, according to Gartner, has something like 32 billion interconnected devices, the Internet of Living Things will be ushered in by real-time sequencing. And right now, we are the first mover and the only company who has these real-time devices. What do I mean by that? During the pandemic, not only were we checking which variant of COVID we had through lateral flow tests, but we were also starting to measure effluent, wastewater, and that holistic approach to measuring everything that makes us up. So our source code, everything that's in us. There are over 1 trillion passengers in our gut that make up our microbiome. We haven't even started to understand how that has an impact on health and disease, and it does. So what's in us, what makes us, what's in the environment, what we eat, pathogens in plants, pathogens in animals, that whole end-to-end holistic understanding of the source code of all living things, which ties into the company's ambition, the analysis of anything by anyone, anywhere, is where this opens up and shifts towards. And we've talked a lot about human health today. Let me give you an example of where this could have a huge impact in industry. Measuring DNA or RNA in real time can have a profound impact on biologics manufacture. And we are doing some really interesting, we're entering some interesting partnerships with experts, domain experts in biologics, quality control, quality assurance, contamination control management during the manufacturing processing of, for example, RNA vaccines. And that is another example of how real-time DNA, RNA information could have a huge impact on the quality, the purity, and the efficiency of that manufacturing process. So this really will, emanate and impact pretty much all corners of our lives in the 5 to 10 year time frame.
That sounds huge. What sort of discussions are going on in regards to the ethical implications about having so much data, so much of this DNA sequencing on file, and the security of all that data? Have you had lots of conversations around that?
So I think that you can well, first of all, as with all new technologies, we need regulatory frameworks to keep up for governments and regulators to provide the right frameworks and tramlines to work in. But you can split the world into sort of kind of 2 distinct domains. When it comes to sequencing humans and interrogating the source codes of human beings and population scale genomics is referred to, the ethics and the frameworks need to be very stringent. At the other end, if we're just interrogating wastewater or somebody's lungs or a fish that's contaminated or has an unusual disease, we're really interrogating the biology of the invading species, not the actual, in the case of lungs, not the human itself. You're just looking at the viruses or the bacteria that are causing infections in the lung. So that's more around communication and education and genetic counseling. If you're being tested in intensive care for a respiratory infection where they scan the sample and look for all the pathogens that could be driving the infection, you're not actually investigating or interrogating the individual's DNA. So there is a distinction there, but there is a lot of education. And one of the things that we're very excited and keen on is getting the MinION in particular, the device I showed earlier, into education. So in exactly the same way as youngsters aren't afraid of using tablets and iPhones and computers because they've always been around, we want to make the understanding of all the different types of DNA from a very early age. And there will be really interesting innovations that will come from having cheap, affordable, accessible DNA, RNA source codes of all living things on the planet.
Okay, Gordon, we've only got a couple of minutes left, and this is going to be a big question to end on, and I've got to ask it. AI, how do you expect AI to be impacting your industry going forward? So
I talked about how the technology works. We inject this piece of DNA through this very small hole, and the output of that is current as a function of time. So it looks a little bit like a New York or a London skyline. And that, inside that signal are the bases, GTAC. So we've been using machine learning AI for deconvoluting these very complicated electrical signals and converting them into GTAC. And in fact, in the last 2 to 3 years, and we use something very similar to what is used in the field of voice recognition, and you've seen the rate at which voice recognition has improved. But that is all underpinned by the revolution in machine learning AI. We have a very strong, vibrant machine learning AI group, and it will certainly has pushed the technology to the point it's at, and it will drive it further. Now, that's in the deconvolution of the signal, but there's also a lot of machine learning AI now going into things like the actual nanopore, so this protein I talked about with a very small hole in it, and how we can use machine learning AI to actually accelerate our understanding and improve the technology further. So it will have a big impact. For us, it's been more about accelerating rational design, whether it's in deconvoluting very complicated signals or creating new biologic sensing systems that improve the signal to noise. It's already been impactful in the last 2 or 3 years, and it's only going to get much more powerful and impactful for our company.
Gordon Sanghera, thank you so much for joining us. It's been an absolute pleasure.
Thanks very much for having me.
Obviously, this is for investment trusts in the UK. I mean, just from an investor's point of view, how important is it for a company like Oxford Nanopore, that's a UK company, to be listed in the UK, or does it not matter from an investor?
I mean, I think our view from an investor perspective would have been that the company obviously has to make its own determination, but in general, companies of that type would be better suited to a listing on NASDAQ. But for some of the reasons that Gordon mentioned around the company's heritage in the UK, the basis of the team being here, it made sense for the company to look at a listing in the UK. But I think we'd be mindful that it is kind of bucking the trend of what has been a very tricky UK market. We've obviously seen some of the changes that the FCA announced earlier in May about simplification of the listing rules, which we definitely welcome. And people will read the article that Peter Harrison wrote last month talking about, our sort of firm view on the thoughts of those changes. I think Nanopore is really bucking the trend at the moment, but is, it's exciting to see where it might go.
Just want to throw in a slight curveball. I've got to mention Theranos. I just wondered whether what happened with the scandal with Theranos has affected investment within the industry, because although they're not the same company, they're sort of related, although by a very long distance.
I mean, Theranos is obviously blood testing done in centralized laboratories. I mean, this is a very different technology, as Gordon said, about going from a centralized mainframe approach to a more distributed approach. Their devices are actually in the hands of their customers, whereas at Theranos, it was they were running the tests themselves in their own facilities. So it's completely different. And to be honest, we don't really see that. I haven't heard that raised in relation to this company, but it's definitely something with these kind of businesses that are doing things that are breaking the edge of technology that you have to be mindful of.
And he's looking to get those devices into as many hands as possible. As an investor, is that nerve-wracking, because do they give up some of their profits by doing so?
No, we see the proliferation of applications of the technology. They need to be very specific about which parts of the market they put their effort into in order to penetrate those larger parts of the market as quickly as they can, to be ultimately sort of profit maximizing for shareholders. But I don't think we see any concern with the way those devices are going to be used. So I think it's really about the question I asked him about education. that's probably one of the greatest barriers they have, is because they have to educate everyone that uses a device. They've not used them historically, and that does take a process of time. So, sales cycles should, shorten it in time, but they're definitely on that upward trajectory at the moment, where they need to educate as many people as possible so that the word gets out there and ultimately commercial traction builds.
And we touched on it towards the end there, with the amount of devices getting in people's hands, all this DNA getting stored, all this information. Have you got any worries about regulation around the sector, around this industry?
I think it needs to be approached with caution. I mean, ethics is a very important topic, both in relation to DNA and artificial intelligence, both topics that we've covered here. So it's something that, the company needs to embrace regulation. And I think from hearing the discussion today, they're very much, of that view. But that's with these kind of technologies, it has to play out over a period of time, and we have to see how people and regulators adapt to the environment in order to make the best use of this technology.
And we spoke about AI at the end there. It's all over the news at the moment, from the barest of warnings to the hype around what it could bring us. How do you view AI's impact on a company like Oxford Nanopore?
So obviously, I mean, as a global private investor, been investing for over 25 years across the firm, we've seen AI for quite some time, and more recently it's been concentrated in the field of generative AI enabled by large language models. So companies like OpenAI, Cohere, that are sort of developing these models that are hitting the headlines. So to make the distinction between AI and what these generative AI companies are doing, I think one of the really interesting things in relation to this company is what they do is so IP rich, so specific, and has been developed over so many years. It's not something that can be replicated like that. It's not something that can be immediately usurped by an AI model that just does it itself. You need the hardware in order to be able to do the analytics. And really, for Nanopore, I see it being more about harnessing that technology in order to improve the existing products, launch new products, and be able to use it to generate operating efficiencies to take the business to the next phase. And that's really about capturing AI rather than being worried about the concerns that it might raise. Yeah,
speaking of AI, I did actually visit ChatGPT before we came on, and I asked it to ask him a question. It came up with, how might the future of DNA sequencing contribute to our understanding of human evolution, population genetics, and the history of life on Earth? I mean, those are big questions to answer.
Huge, and I would say Gordon would know how to answer this far better than I can.
I thought you might say that. Okay, so one final question. Where do you think overall the market might be in 5 to 10 years? We've heard from Gordon. What do you think?
Commenting on the sequencing market overall, or this stock in particular?
Let's say the market overall.
I mean, as I said, I think we're seeing the proliferation of applications of this technology. It is about educating people. It is about converting them, proving the ROI of this technology. And I think from what we've seen from some of the case studies that the company's talked about, that once they do embrace the technology, there are very fruitful potential gains from using previously unseen data. So I think we just need to see where that journey takes us. And really, it's a product of decisions that the company is making right now and will do in the future. But I think we're confident that the company is at the leading edge of what it's doing, and that there's a pretty bright future ahead of it.
Yeah, it's an amazing one to follow, isn't it? Because it's not just about us human beings, and or the diseases that we might catch. It's about food, what's the pathogens that might get into food. It can be on war zones, stuff like that. Everything.
Exactly, things that people haven't thought about.
Yeah.
And until they get a device in their hands and are using it and seeing the results, that's really the proof point.
Yeah, brilliant. Harry Rakes, thank you very much for joining us.
Thank you.
Cheers.